Welcome to the Over the Hump Podcast. Your gateway to the world of the iconic Brahman breed of cattle. At BRC Ranch, our passion for Brahman cattle runs deep. Join us as we delve into the heart of this extraordinary breed, unveiling its rich history, exploring industry trends, and sharing captivating stories that define the American Brahman. BRC Ranch stands as the pinnacle of Brahman excellence in America, boasting a decade-long legacy of champions and housing the finest Brahman bulls worldwide. It's not just about cattle, it's about a legacy that reverberates across ranches and hearts. Brought to you by BRC Ranch, where the Brahman legacy thrives and stories are woven into the very fabric of ranching. This is Over the Hump.
Hey everyone, Rachel Cottrell here, and I am totally geeking out over this episode that you're about to listen to of Over the Hump because it is so good, especially for people who really love animal science and really love research. I sat down and visited with Dr. David Riley, who's a leading animal geneticist from Texas A&M University, and he just shared so much good information about Brahman genetics. And I tried to put together what I thought are like the most common genetics questions that people ask. And so some of the big topics we talk about are horned polled in Brahman, hair color in Brahman, like red versus gray, speckling, black, all of that kind of stuff. We talk a little bit about genetic defects. We talk a lot about heterosis. And I even asked the question of, does overaging cattle for the show ring affect EPDs? And you'll have to wait till the end to hear that. But it is just an amazing interview. And I'm so thankful that Dr. Riley would take the time to share his expertise with us. And so thankful to Texas A&M Animal Science for the continued priority that they put on Brahman cattle research. And so this episode, I know people are going to love, whether you're here in the USA or you're international and you're just craving information about Brahman genetics and genetics research. This is one that you are going to love. So hope you enjoyed this episode of Over the Hump. Hi, everyone. Welcome to this week's episode of Over the Hump Podcast. So I am your host this week, Rachel Cottrell, and I am really excited to introduce this week's guest, which is Dr. David Riley from Texas A&M. So welcome, Dr. Riley.
Good morning, Rachel.
Thank you so much for joining us. I want to do a quick little introduction of you because you're so smart and amazing. But for those who may not know Dr. Riley, he's a professor of animal breeding and genetics at the Department of Animal Science at Texas A&M University. And he teaches 3 courses, which are so smart I can barely even say them, but quantitative genetics, genetic prediction, and advanced quantitative genetics. And so he's been at A&M since 2009, but before that he worked for the USDA at the Agriculture Research Station in Florida. And so we are so thankful for you to spend your time with us today, Dr. Riley.
Well, gosh, the honor is mine. And, that was a nice introduction. Thank you so much.
Well, and I forgot, honestly, the most important part. I mean, not the most important part, but you love Brahman cattle, right?
Yeah, for sure. That's our work. That's the cattle of our customers, of our research, for sure.
Yeah. Did your first experience with Brahman start over in Florida?
Really, my first experience was in graduate school. Yeah, with Dr. Jim Sanders at A&M. So that was my first real working with Brahman cattle, but definitely in Florida, I got the full dose there. Yeah.
Yeah. Do they do research on Brahman cattle at Brooksville? I mean, that's probably a dumb question, but.
Well, they did. Okay. Brooksville has closed. So about 2010, there was pretty regular pressure to close small USDA research facilities, and that one was finally closed completely in 2012. Yeah.
Okay. All right. And then, of course, you do a lot of work with Brahman cattle. So maybe before we get into my questions of here, would you tell us about how A&M, Department of Animal Science, uses Brahman cattle, or what they do with the Brahman cattle research?
Yeah, definitely. I'm happy to. Today, we have a big project involving Brahman. We prepared for this project for probably maybe 10 years to get the cattle lined up to produce these. But the newest project we have is to investigate the genomics of heterosis using Brahman and Angus. In order to do that, then we've got several groups that we're producing simultaneously, that is, as contemporaries. And genetic research, that's really important to us, is that they be produced simultaneously. So these will be 100% Brahman, three-quarter Brahman, one half Brahman, one quarter Brahman, and all Angus. So we've got 1 year of calves that are being weaned this week at McGregor, the AgriLife Research Center at McGregor. And we will do this for 5 years. And all the females from that project will go into their own cow project where we're investigating and using our knowledge of heterosis and then genomics, trying to mesh those two things together. The really great thing that I like a whole bunch is after I had put this breeding scheme into place, my colleagues at Clay Center, Nebraska, United States Meat Animal Research Center, contacted me, and if you probably remember, but they use 18 different breeds of sires, and they tried to sample about 10 of the most used sires in every breed to produce and to breed to their 6,000 cows. Well, they wanted to do that with us, and they are doing that at Beeville, by the way, the AgriLife Research Center at Beeville. But I said, I'm only interested if we just do Brahman and Angus sires. So they agreed to that. So we're tied very closely to the research center at US Mark in Nebraska, and they're our friends, they're our colleagues. So we see a lot, and of course we're tied very closely to Beeville as well, because Beeville will be using some of the same sires. And so really happy about this particular project. We also will be able to validate with the big Brahman Angus herd it's a crossbred kind of herd at the University of Florida. So they do a little differently, like they've been crossing wild ones across all different possible proportions, and therefore their DNA on both sets, like each animal has two sets of DNA, one from their father and one from their mother. Theirs is a complete mosaic of Brahman and Angus. Clay Centers, all of those animals will be half-bloods. They'll be sired by Brahman and sired by Angus, the cattle that we'll combine with. And their maternal genome is the complete mosaic. And then ours is entirely structured and deliberate to estimate heterosis. So you see what a dangerous question it is. Ask somebody who's in science what they're working on, right?
Yeah, that is amazing. Well, I want to ask so many questions, but I don't know the right words because it's so over my head. But when you say the genomics of heterosis, is that like how much hybrid vigor each contributes, or?
Yeah, so what I think, okay, and there's been a little bit of work done in this in Bos cattle, and my colleague, Dr. Relika Matescu at Florida, also started into this, but she's retiring, unfortunately, this year. But we think that probably, so when so each animal has two copies of its entire genome, right? One from their father, one from their mother. And we can go through and compare, and where they have the same nucleotide or allele. Remember, there's four nucleotides in genetics: A, C, G, and T, and where they're the same, we call that homozygosity. Inbreeding causes them to be the same. It causes them to be homozygous. When they're different, we call that heterozygosity. So we know, I mean, that heterosis is due to heterozygosity, but we think that might not have to be genome wide. So what we want to do is find the places that seem to be absolutely essential for heterosis expression in these crossbred cattle. And of course, the traits I'm most interested in are those related to mature cow performance, right? Also their puberty as well, but more so their mature cow performance. So that's the notion, is maybe we could find a place. So we have a reference map. So we number chromosomes from one, the largest, to the smallest, which is 29, and then the sex chromosomes. The X chromosome is enormous and the Y chromosome is a little bitty. So, but from 1 to 29, we have a pretty good reference map, a reference assembly. So we know addresses of confirmed genes. We know places. So we might be able to find a place like, for example, we always have found results when we're doing genome analyses on chromosome 5. So I wonder if we were just heterozygous at this region on chromosome 5, is that sufficient to produce the heterosis in the mature female? So that's sort of the notion. Obviously, we'll look at breed of origin, like we know that this part of the genome, we'll be able to trace that specifically. This part of the genome is from Brahman, this part is from Angus. Where do we have that breed of origin difference, and do we need to have that at specific places in the genome? So it might allow us to exploit heterosis and then select really hard for other traits like carcass traits or various things on the side without disrupting that heterosis that we know is the most important genetic. To me, Rachel, heterosis is the most important genetic effect for beef cattle production. And it'll be the one that feeds the world. And your cattle, a big role of theirs is in the production of animals that will express heterosis, right? Those F1, Brahman Hereford, Brahman Angus, yeah.
Oh, this is going to be such a good podcast. Oh, this is going to be so good. I do have I do have another question about heterosis. And okay, I got to say, I'm like one of these people that I don't know very much about genetics, but it fascinates me. And Brandon and I talk about this a lot. Our pedigrees at BRC were really tight with certain bloodlines, and we started noticing they were getting really tight. And so maybe a few years ago, we went to Well brothers, who are mostly Imperator bloodlines, and we went to Parton Ranch, which is Emperor, but a lot of Florida bloodlines, you know. So they're still purebred Brahmins, but entirely different lines than what we have. We felt like we saw like within breed heterosis because maybe they were just so different. Is that possible?
Yeah, it really is. And you're really you guys are really sharp, and I would love to sort of document your evidence for my class. So a colleague of mine who has retired but kind of is a consultant now, his name is Dr. Mike McNeil. He also showed that in Hereford. So what he did was there were two lines of Hereford cattle that were very distantly related pedigree. And when he crossed them, he got a heterotic effect. So I'm not surprised, and I would love to have that example.
Well, yeah, I need to share it with you, but like one of them that is very obvious. So the cow line that we were working with was not known for its fertility. I'll just say that. But the Parton bloodline, as you know, is just a century of fertility. And I know fertility is low heritable, but the Johnny Brahman bull that we made from that, he's like passed his fertility test at younger than any other bull we've ever had. And that's just one example. Like all the daughters from that, they started, you know, they got bred early, and we really believe that that is what happened. They were so far apart, even though they were registered Brahmin, it just clicked so good.
Oh, I'm sure. I'm sure it's what happened, and that's a really beautiful example. I love it. I would like to, because our students they need to have it in. I always present it in Hereford terms, but they really need it in. I like for A&M students to be super knowledgeable about Brahman, and my Brahman examples would be really good. But yeah,
Well, let's get into our actual topics that we planned on talking about. And for our listeners, what I try to do is maximize this time and think about the most common genetic questions that we hear in the breed, and then toss those over to Dr. Riley and get his take on it from a genetic standpoint. So the first one would be the genetics between horned and polled. And, you know, Brahman is primarily horned, and but polled is gaining popularity. But we as breeders find that is I don't know if volatile is the word, but especially at the beginning, sometime you would breed a polled to a polled and you might get a horned. But my thought, like my knowledge of shorthorn, is that always polled is always dominant, and you're always going to get a polled no matter what. So can you just walk us through big picture genetics behind Brahman horned versus polled?
Okay, well, I'll sure tell you what I understand and what we teach in my class for sure. Exactly as you said, polled is dominant. Horns are recessive. So that means that of the two alleles that they have, an animal has to have both of those to be genuinely horned. Now, there's a third phenotype we would call scurred. So scurs are similar to horns. Like they may look like them. In general, they're not really attached to the skull like a horn is. So they're, and in fact, like, and size doesn't even matter. They could be as big as your fingernail, or I'm convinced also even might not even notice them. They might just be a bump or something, or they might be big and pendulous. We have one cow; she's a crossbred cow at McGregor, but her skirts just swing in front of her face all the time, you know. So skirts is it's a different phenotype slightly. So yeah, so we know that polled is dominant, but I think first I would say sometimes the skirts can be missed. So you might have, and by the way, in order to be scurred, we think that an animal must be heterozygous. That is, they must have one dominant allele that would result in polled, but they also have a horned allele. But there's a separate place in the genome that may be responsible for that generation of scurs. And my colleague, Dr. Claire Gill, this is one of her. She's had a student that was super good who they're working on trying to finalize a document on this. But yeah, it appears to be a second gene that influences the expression of scurs. So polled then, there have been at least, so a lot of times when you have a gene, some mutation causes that allele to result in the phenotype we see, like polled. Well, there have been four separate ones. So we know where polled is. It's on chromosome 1. That is the biggest chromosome in cattle. And it was discovered in the early 1990s by a famous Belgian geneticist. So, but there are four different alleles that will convey the polled phenotype that we know of. And they're probably, as is the case with most genetic stuff, we probably only know a very little bit of the whole truth, right? So, but we aim to know more of that, right? So there is one called the Celtic allele. So that is the allele that's been noted in Angus cattle, and it's also been noted in polled British cattle. There's another one that is Frisian, so they call it the Frisian allele. It will convey polleness, and it was discovered in Holstein. There's a third one that was discovered in it was some German geneticists, and it was in some different breeds, and I'm sorry I can't come up with the name of it right now. I think it's the Mongolian version, but they were working on maybe Chinese cattle and somewhere over there. And then finally, in just recent years, they discovered a variant in Nelore cattle in Brazil. So those four alleles we know have existed. So yeah, so selecting for pole, I'm not surprised that you might see some variation there because especially I think skurs get misinterpreted as horns sometimes. And we might not always notice them, and especially at young ages. I try to get horn phenotypes on all our cattle, and I'm always shocked at how it changes as they age, right? Because I think they're horned and it turned out to be scurred, or I call them polled and they turn out to be scurred later. So it's difficult. So that's kind of the genetic basis for polled and horned and scurred today. Address what you were asking?
Yes, that is so good. So, okay, a common thing I've heard long ago is that, and I don't even know if this is an urban legend or just if it's true or not, but there are some countries that that won't allow a polled Brahmin to be registered. Or I don't know if that's still true, but it definitely was true, years ago, like Mexico wouldn't allow a polled Brahman to be registered as American Brahman. And again, I don't know if this is true or not, if it was just rumor, but they said the rationale was because they think it would have had to come from the Angus breed at some point, and so therefore it's not a purebred. I don't know. They may have. I think they might have changed that recently, but then when you said they've noticed one coming from Nelore cattle in Brazil, which Nelore is a base of the American Brahman, do you think do either of those make sense as where it came from Brahman?
Yeah, I have some opinions on that. So you know as well as anyone that the American Brahman was developed from sort of a Bos Taurus base. So I'm not surprised. I don't know of a bunch of Brahman cattle that have been genotyped for at polled and conferred which one of these four alleles they have that conveys the polled phenotype. But I would suspect the Celtic version, that is the one that was discovered in British cattle, might be in Brahman. It is not unreasonable also to think that the version that, by the way, the allele from the Nelore cattle they call the Warani, which is like some Indian tribe or something down there. I would not be surprised if that allele was in Brahman as well. And plus, we've discovered four alleles, four mutations that result in this phenotype. It seems pretty reasonable to me that there are more, right? Because you'll know, for example, the double muscling phenotype in cattle. It's due to deletions in one gene. That's a really good discovery. But they found 9 different versions of alleles that convey the double muscling phenotype. So I think it's pretty likely that more alleles that resulted in the dominant polled allele are out there. But it is reasonable that it came from Bos Taurus. That is specifically probably not what I would think the Frisian allele, but probably British sort of deal. That's not out of question, and it's also not out of question to be out of the Nelore as well. One thing I didn't tell you, this is pretty good too. This is old stuff, but in the 1930s, these guys in Africa bred some Bos Taurus bull Angus, I think, to some Bos Indicus, and of course they weren't Brahman. They were Brahman or one of those African versions of Bos Indicus. But they thought they found, like, all their females were polled. Then their males, they had horned males, they had scurred males, and they had polled males. And so they theorized, and you may have heard this, they call it, maybe it would be the African horn gene. So they thought there was another place influencing the generation of horns, and it was sex influence. So yeah, that's pretty interesting to me too. That's never been confirmed, and we're a long ways. We haven't really squared that away, and nobody has really documented the evidence of that African horn gene. But you may hear that in cattle circles sometimes.
Elevate your herd with the finest Brahman heifers from BRC Ranch. Perhaps you need to replace ranch heifers, or you're sourcing elite show heifers and donor prospects. BRC Ranch understands your passion for Brahmans. We feel the same. Building a strong foundation is essential for long-term success, and we invite you to see our exceptional Brahman heifers in person. Schedule a visit to our ranch by going online to brcottrare.com and selecting a suitable time slot. That's brcottrare.com.
Well, you said scurs can be missed. And another fun little urban legend we have is that one of our most popular bulls, Noble, his mother was smooth polled. And she produced several progeny that were smooth polled, but Noble himself, we registered as horned. But he started throwing occasionally some polled calves, and we got to thinking, was it possible that he was scurred and we just missed it? Like, when we were dehorning, we went back and we have found some pictures of him as like a baby calf and kind of looked at his skull shape and stuff. And we ran a horn pulled test on him and it came back inconclusive. But that's something that we've always just had in the back of our mind. Like, is he really maybe scurred and we just missed it?
That's how I'm thinking when I hear you tell me. Yeah. Yeah. Super interesting, huh?
I know. Well, let's move on to our next topic because we could just go all day. But the topic I want to talk about here is Brahman hide color and like skin pigment. I guess the inheritance of coat color in Brahmins. 'cause it's been a while since I took Dr. Sanders' class, but I remember we would talk about, he would talk about the nose. Like you might have a dull colored nose and a shiny nose, and the difference in their ear color, the pink ears versus the black ears, or the black on their testicles. Can you give us an overview of a quick crash course in that?
Yeah, I like this. We demand this from our students every semester. Yeah, so it's an interaction among three different genes that result in what we see in Brahman cattle. The first one is we call it the extension locus. We know what it is. It's melanocortin 1 receptor, but it extends black pigment throughout the coat, and it is dominant. So when you see an Angus animal, usually those animals have that dominant allele. There's a recessive one too that results in red coat color. And when you see red Angus, they're homozygous. They have two copies, that is, of that red allele. Then there's another allele that we call kind of a wild type allele, and that's what Brahman usually have. So they usually have two copies of that. So when they have that background, there is another gene that we have not documented, and we in my class call it the B locus. And when animals are homozygous large B, then they will show that black nose, feet, and tail phenotype, like you're describing. Usually dull black nose, right? That kind of thing. But when they're heterozygous, that is big B, little b, there's only two alleles at that one. We're surmising. By the way, I would add that he was my friend and colleague at University of Florida, Dr. Tim Olson, did not agree with us on this.
Oh, okay.
That's funny. You'll find geneticists that don't agree with each other. Imagine that, right? Then there is another gene called agouti, which we do know exists, and we know where it is. It's on chromosome 13 in cattle. So agouti is one that puts stripes on mice. It has versions, we call those homologs, or homologs, that means another version of the same gene in different species. So it puts stripes on mice. It's responsible in crossbred cattle. We get a brindling phenotype from a certain genotype with this at the agouti. But when there's homozygous recessive and heterozygous at the B locus, then we get the Brahman cattle that have the shiny black nose, the black tips of the ears, black bases of scrotum, mainly those kinds of phenotypes. Yeah, so that's the genetic basis that we teach. It's not confirmed. I mean, we have not experimentally described it, and I can't even tell you what Tim thought. Tim's been retired for 10 years, and I'm sure I had it at one point, but I don't have it now. I'm just pretty convinced on ours we've seen enough cattle.
So if a breeder has a Brahman that, let's say, is a gray Brahman, but it has like a dull nose or even a pink tint to its nose, if you bred that to a Brahman that had the dark shiny nose and the black tipped ears, should that fix the problem?
Well, I mean, you have a chance of it, right? I mean, because you've got two genes working in there, and you've got three genotypes at both genes, essentially two genotypes, because we think there's dominance at both of those. So essentially two. So you have four possibilities. So you're looking at, there's some probability to it, but it is not certainty to establish that one, right? Which is probably why you see some variability. So Brahman should probably all either be, there would only be one genotype at B. So it might be an interaction of three genotypes. But still, even in that case, where you've got maybe not a quarter probability, but now it's a little higher than that, maybe a third, So it's not surprising there's variability. Yeah.
Yeah, that makes a lot of sense because, like I said, I'm kind of striving for that black, shiny black tips. And we have some flushes that I think of that are the exact same mating, but there is variation within those calves, like in their nose color or having the black tips and stuff, even though they're the exact same mating. So that really explains it to me because like you said, you have different possibilities of what it could be.
Yeah, and they're with equal probability, basically.
So red and gray Brahmins, this kind of goes back to it too. I've always heard use the rule of thumb that if you take a gray Brahmin with the black tipped ears and breed it to a red Brahman with the black tipped ears, the calf should come out as red. And so is this those same genes you were talking about?
So this is different. So there's a different gene responsible for gray coat color. And in cattle, it's the recessive allele. So red is dominant. The non-gray, we wouldn't say red, we would say non-gray is dominant to gray. So think about that. If you bred two gray Brahmins, you would definitely get gray offspring, right? But breeding to a red Brahmin, depending upon their genotype at the graying locus, if they're red, they clearly have the dominant the dominant allele, but they might be carrying, it's reasonable that they would be carrying the red allele, so half their progeny would be gray and half of them would be red if they were heterozygous. Yeah.
Yeah, that is very cool. I also like how they just subtly call it gray versus non-gray because I'm a gray breeder first and foremost.
Yeah.
I like that. Okay, what about speckles? How do you get that? Or is that a dominant? You see that a lot in reds. I even see that sometimes, like Captain, our bull, he's not speckled by any means, but he has a little bit of speckling under his neck. And he's a darker pigmented bull with like jet black, skin. Where does speckles fall into this?
Yeah. So there's one gene called KIT, and it's responsible for spotting. And there are probably 7 or 8 alleles. And these alleles are in general, now non-spotted is dominant, okay? But in general, the others are all sort of codominant. That is, you'll get partial expression of both. So there is one allele we think is responsible for speckling, and it is codominant with the other alleles, and even with the solid color. So that's why you may get some partial speckling and location of the body. I can't really. Nobody would. And this hasn't really been documented either. It's just what we've observed, But 2 copies of that speckling allele at this KIT locus, which is responsible for spotting. For example, there's an allele that results in that 2 copies of it results in the Hereford pattern. Okay, 1 copy of it results in bald faces, right? So you combine the speckling allele with any of those, and you get generally along the edges of the other whites some speckling, but it could occur anywhere. And some of those really interesting looking Gyr cattle from Brazil or other places that have 2 copies of speckling, you've seen those, they're speckled all over. And that's pretty interesting too. But yeah, it's one gene, but 6 or 7 different alleles that are codominant. That means they each evidence themselves in what we see in the color, partially.
Last question about Brahmin color. So there's like this new trend that is black Brahmins, but if you see them, they're not. So black and Brahmins like have been around in red Brahmins for a long time. You would have like a solid black almost bull, and there's dark pigmented grays. But some of these new quote black Brahmins, they are black, but they're almost brownish black. It's like not as firm or strong black. And so some of them do have like black ears, but some of them have like white hair around their ears. It's like their skin are not black, but is there any tricks or information about black Brahmins?
Yeah, so I'm just surmising here. I'm trying to guess, right? But I have the first Brahmin I saw that was completely black was in Rockhampton, Australia in 2008. And I have a picture of it still, and it was a cow. So think back to the shiny black nose, the black tipped ears, black base of the scrotum, of course the black switch and black feet, right? So in that particular genotype, what I think I have observed, and you would know this too, but for example, I'll have some female. For a while I thought it was sex specific, but I don't think it's sex specific. But I would have some animals that would seasonally darken up. You know what I'm saying? Especially at the front, like on their head and on their rear end. I'd have a cow who had been really light colored during the summer. All of a sudden in the winter, she was very dark in the front and the rear end. And it was always those cattle with the black rimmed ears. So I think there's a lot of red Brahmin that are that that genotype, right? And so what I think we see is not melanocortin 1 receptor, that is the gene that conveys black color to Angus cattle. It is not that allele, because you're exactly right. But I think it's an excessive expression of that darkening thing, right? But then that's just all speculation and no science to confirm that. And probably you guys have as much anecdotal observation on that as I do. But do you have cows that darken up and bulls that darken up?
Oh, oh yeah, absolutely, for sure. I love it too. I mean, and I love that. And doesn't that somehow go back to that born red and turn gray hair color? Like, his captain, when he's almost jet black and he darkens and lightens a lot, but when he was born red and turned gray. So that's a whole nother fascinating thing about Brahman.
I think it's an extreme version of that darkening up thing.
It just confirms that Brahman is the world's most unique breed.
Yeah, I mean, that's what I suspect anyway with it.
Oh, that was great. Okay, so I have two more topics I want to jump into still if we have time, but the third one is genetic defects. And we hear about this a lot in other breeds like dwarfism or like the TH and PHA in club calves, and I know like there is a condition in Brahman called Pompies that kind of breed, but in large part, it seems Brahman has been able to avoid genetic defects. Do you have any insight on that or thoughts on how genetic defects do or don't affect our breed?
So, well, it's like this again is another great topic, and it's part of our class as well. But because where do you see most of these genetic defects? What breed do you see them turn up in usually?
Is it Angus or Hereford?
Yeah, I think so. And I don't think it's because they're inferior, right? I think it's because they're just so widely used, right? So, I mean, like for instance, and I don't want to denigrate them, they handled it so perfectly. But Gardner Angus Ranch with GAR Precision 1680 and that bovine multiplex arthrogryposis or curly calf syndrome, man, that thing just shot off, right? All of a sudden it was just, but that bull had over 10,000 sons and daughters in the Angus breed. And so it was just inevitable that they would be bred together at some point or close relatives would be. And again, the Gardner Angus Ranch handled that situation so perfectly. And they were a model for the rest of the cattle industry with regards to that deal. But I think it's probably at this point, you just don't have the population size that is going to generate it. I think they're there. I don't think that, for example, curly calf syndrome is limited to Angus. I think that it could be expressed in other breeds, but we saw it in that breed because of the mating structure. Although you have some mating structure and you definitely have some line breeding in the Brahman breed, it just you haven't seen as many things come up. At least that's sort of how I see it today.
That makes perfect sense. That explains it. I was just thinking it was because our breed is so strong. But yeah, thank you. That really explains that. Let's talk a little bit about heat tolerance genes because I think this is one I know I personally would like to hear a little bit more because I'm seeing some other breeds like Boss Taurus breeds putting out like slick hair measurements or EPDs or stuff. And I think to myself as the Brahmin promoter, well, we're the ultimate slick hair breed. But so I know there are genes associated with heat tolerance, but is Brahmin a unique gene for heat tolerance, or is the same gene that some of these other breeds are talking about now?
Well, so I would say there's a lot of genes involved, right? Really, the only one that's really been confirmed has been what you referred to as the slick gene. And so that's a very unique, it's also a dominant allele that conveys that short slick coat like you would see in particularly Centipole cattle or the Criollo cattle from Colombia. Lots of them have that as well. So it's great because Tim Olson started this, same guy I referred to a while ago. He started maybe 15 years ago trying to assimilate that particular allele from Centipole into Holstein herds in Florida. So it sort of took off. It's been confirmed as a definite gene that we know. That's the only one that I really know specifically to convey adaptation to hot conditions. So I think probably the rest of them, it's lots of genes. And so whether it conveys, we can all argue about whether it really conveys tropical adaptation to the extent needed. But the fact is, so I would also say Brahman cattle don't express the same way. So their hair coat is not the same as a Centipole's, okay? That dominant short slick phenotype. But they're certainly adequate, and they're adequate in a bunch of other ways. So I suspect there's a multitude of genes that are responsible for this, and a ton of research that supports that. Especially that's probably one of the reasons that Brahman is in our industry also, in addition to heterosis, which is the most important one, is they convey adaptation to a crossbred animal, right? And we documented that in Florida too. We documented heterosis for core body temperature in crossbred Brahman Angus. And that means heterosis, of course, is the difference of the crossbred from the average of the purebreds. And so that's a really important contribution. But I don't know of genes that have been specifically identified that convey tropical adaptation other than SLIC, other than that particular gene. And I don't know, I think the jury's still out on that one, whether it's going to really sufficiently convey to Bos Taurus animals the ability to get along in South Texas or in the southern United States.
Mm-hmm. Oh, that is so fascinating. I want to switch gears here a little bit because I feel like we've been talking like really deep science stuff. And this is my last question I want to ask, but it's more of like street talk question, I would say. And that is EPDs. So we hear this a lot in the Brahman breed that you can't trust EPDs, or some people, live by EPDs. But the street talk part of it is most people refer to or incorrect, false information as the reason why they say you can't trust the EPDs. So maybe it might be a person who doesn't actually take a birth weight and just eyeballs it, or a person that just turns in 70 pounds for every calf that they register, or even as it relates to the show ring, people who change the birthdays on their animals for show ring purposes. Do things like that affect EPDs accuracy?
So I think so, yes. Anytime it's a mathematical outcome, right? I mean, so whatever we put into these equations to estimate EPDs for animals, if it's inferior information, then that the backside of it is it'll be inferior on the backside too, regardless of what the accuracy measure that the breed association reports is. It will be no good, right? So all the situations that you mentioned there are definitely things that compromise the effectiveness of being able to select with EPDs. Okay, so in that sense, the association members, we all rise together or we all drown together based upon what we all do here. So it is in the best interest for everyone to go, we're going to be legitimate on this and we're going to give real numbers, because real numbers generate better predictions, right? More reliable things that we can use. Now, the system generates an accuracy for these EPDs that is not related to that. It doesn't pick up whether the information is false or not, right? It just the information that's put in there and it generates an accuracy. So let's not confuse that numerical accuracy that the breed association reports with how good the EPD is. Is it real? I mean, one of my favorite statistical quotes is, it was in terms of a statistical model, but it explains EPDs too, and we teach this to our kids as well, is that all of them are wrong. They're estimates, right? some of them are better than others. We get better with more information, and we get better with more accurate information, right? More information visibly changes the accuracy that you get from the breed association, but fixing those other things doesn't reflect itself in the accuracy that they report, but the number is better.
Yeah, yeah. That explains it so well. Thank you. Yeah, that's great because I think that's a question that a lot of our breeders would ask. So, this has been great so far, and just as we wrap up, I want to ask you, is there any big topics coming down the pipe that you think are, the next big thing in purebred seed stock breeding or cattle breeding or just general genetics for cattle?
So the big topics for us now that everyone wants to be a part of is methane emissions.
Oh, yeah, yeah.
I'm not convinced on that topic. I'm not working in it right now, so it's hard for me to pronounce judgment on it. One thing I found too is that scientists, they're not really interested in anything but their own research, right? So I'm trying to be mature and pay attention to this, but the public relations is sort of demanding that the beef industry address this, and whether it's valid or not, address it. And some of the ways that we're doing it are pretty difficult, right? And so I'm not totally on top of it, but I think that's a big topic. Another topic that has just blown me away, we sent a kid to Australia who you know pretty well this summer, and the biggest cattle breeders there have over 300,000 cows in their company, and they're really going hard Wagyu, which I never thought this would happen, but they are really, really pushing Wagyu to the extent that I think it's going to happen over here too. So I don't know. I mean, the Wagyu cattle I've been around were never great performers, and I haven't been around a bunch, but that's a big trend in our industry that I see from a research perspective is how are we going to incorporate that breed into North American beef production. So that's pretty interesting too, I think. There's a large crowd in our group that always are working on EPDs and how do we make them better and closer, and mathematically how do we make them more precise. And this is focused on the accuracy value from the breed association, not whether there's genuine information behind them or not. So, but there's always people working on that, and our ability to compute just exponentially has taken off. I mean, so pretty soon we're going to be able to incorporate huge amounts of sequence when we estimate EPDs, and those should make numerical accuracies reported by the breed associations go up for sure. So that's always a big hot topic. There's a few prominent places and a few prominent breeders that are really trying hard to to use gene editing. That is to put it that's the latest greatest. And our problem is we don't have any really big genes that are responsible for most of our traits are controlled by a bunch of genes that really we haven't characterized. So I'm aware of people who've edited in the slick, the dominant slick allele into cattle. And I'm also aware of course, editing polled into horned Holstein. Yeah. And so that's going to continue to be an area of exploration. One of the really coolest projects I heard of was these guys found a way, like in pigs, they could they could stop spermatogenesis in a particular animal. Then they could come in and they could edit a gene that would allow them to bring in donor semen into these are boars. And so then they thought they could take these to places where they couldn't use artificial insemination. They called it surrogate sires. You can look it up online if you want to. Some guys at Washington State were working on it. I thought that was a pretty clever manipulation of genetics. And then, of course, there's the famous cow Ginger who had the bovine viral diarrhea virus receptor edited in, and she's still at Clay Center. We saw her in May. We were there in May with our colleagues there, and we all had our picture made with her, right? Deer cow, and she's super gentle deer cow, but all of those things are really in their infancy, and it's going to take a big paradigm shift for us to be able to implement them and exploit them at the commercial level, so those are the big things that I'm aware of. And in cattle genomics, I really think the confluence of cattle breeding with genomics, obviously we designed our project and proposed it and did it the way we're doing it because we believe that's a valid, fruitful direction to go. So I'm a little biased toward my own projects, right? So I think that's a good direction too. And obviously, like in the cattle business, it's the same with geneticists. Not everybody agrees with me, So imagine that, right?
Well, I find it funny that even the world's smartest geneticists take selfies with cows. If you're taking a picture with Ginger.
Well, like my dad would say, let's not call for a vote on that world's smartest thing, right?
Yeah.
I wouldn't like the outcome of that vote. So you're kind to say it.
That's awesome. Dr. Riley, this has been so good. I just can't thank you enough. I mean, so as we wrap up, I do just want to say so sincerely thank you for taking the time to answer these questions and share it with like beef producers like me. On the podcast, we have listeners worldwide, a big global following, and I know this is going to help a lot of people worldwide, help them understand. And also just thanks to Texas A&M for, continuing to prioritize research that really helps producers, like grassroots producers like us. It was just awesome. You're the best.
Well, I love hearing that, and that I appreciate the two of you just a whole lot, and love to bring my students there. It's so good for them to see you guys and to hear what you're doing, and the chance to talk to you is very flattering. Thank you for inviting me.
So if anybody is listening and you kind of want to learn more about some of these projects, you can go to animalscience.tamu.edu and also look up the AgriLife Extension Service. They have a lot of really good research and extension going on. And again, Dr. Riley, thank you so much. We really appreciate it.
Thank you, Rachel. Hope to see you soon.
You've been listening to the Over the Hump Podcast. Our passion is Brahman cattle, and it runs deep. And this show is about its rich history, industry trends, and stories that define the American Brahman. We hope you've enjoyed the show. If you did, make sure to like, rate, review, and subscribe. We'll be back soon. But in the meantime, find us on Instagram and Facebook at BR Cottrare. See you next time on the Over the Hump Podcast.